Hello, welcome to the annotation and database session on the last day of the Biodiversity Genomics 2020 conference. We're excited to have you all here. We have a list of 6 speakers. Now, when I say we, I mean myself, Françoise Thibault-Nissen. I am a staff scientist at NCBI, and my focus has been on the production of whole genome annotation that go in RefSeq for eukaryotes and prokaryotes as well. And I am co-chairing this session with Paul Fliczek, who has led the vertebrate genomics effort at EBI for many years and is now the Associate Director of Emborn EBI, responsible for data resources and other activities there. So without further ado, I want to introduce our first speaker, who is Fergal Martin from Emborn EBI. He will be talking to us about scaling and improving and deploying ensemble annotation. Take it away.
Hi, my name is Fergal Martin. I'm the coordinator for vertebrate annotation for Ensemble. And today I'm going to talk about scaling, deploying, and improving Ensemble annotation. So what is Ensemble? Well, Ensemble is a platform for genomics that enables and accelerates downstream science. We're most widely known for our genome browser, and you can see a screenshot of it here on the right-hand side, where we provide gene annotation, comparative genomics, variation, and regulation data. But there's also a bunch of other ways to interact with Ensemble, like our FTP site, our REST and Perl APIs, our MySQL dumps, and our Biomart data export tool. So the first topic I wanted to talk about today was scaling annotation. And as I'm sure you're all aware, there's all these really cool projects like the VGP, Darwin Tree of Life, the Earth Biogenome Project. And, effectively, they're generating all these really high quality reference assemblies. And we need to do annotation on all of these because a lot of the downstream analysis is going to be based off the annotation. And we've been thinking about this for several years in Ensemble in terms of preparing our annotation pipeline to scale with all these assemblies. Historically, it took about 4 to 6 months of a person working full time to annotate a single species. And through a process of kind of automation, incorporating newer technologies, optimization, parallelization, we've sped that process up massively. And this is a kind of a very high level overview of how the annotation pipeline works. And the main takeaway from this is primarily we like to annotate using transcriptomic data because it can be more accurately aligned to the genome. It's more representative of the actual genes that are active in the genome. But where the transcriptomic data is either fragmented or just not present, we use homology data to do gap filling. And this is a kind of an idea of how long a typical annotation would take. I picked Atlantic herring because it's, a sort of an average genome size, and it had a sort of average amount of RNA-seq data as well. I think it had something like 9 tissues worth of data to do the annotation from. And in this case, it took about 3.5 days to do the annotation, which is obviously a massive improvement from the sort of 4 to 6 months. If you have species that are, have bigger genomes or they have an awful lot more transcriptomic data, that pushes the annotation time up, but we're still talking, somewhere on the order of like several days to maybe a couple of weeks in extreme cases. So, that's kind of great that the annotation itself is faster, but now what we're looking at is how can we actually get it into the hands of users faster? So that's scaling deployment. And historically, we've been a bit slow to update. We do 4 updates a year, so a 3-month release cycle. And the speed of the gene annotation process, as I said, has dramatically increased. But this is effectively, as a result, there are lots more species going into each release, and this puts pressure on other aspects of the project, particularly like the comparative analyses where we do all versus all comparisons, and even just the web footprint. So to solve this, we've been building a new site. It's called Ensemble Rapid Release, and it's live on rapid.ensemble.org. We launched it in early June, and unlike ensemble.org, it doesn't just house vertebrate species, it also houses all eukaryotic species. And we decided for this, we were going to start really simple with, focusing on this reworked, much shorter release cycle with just the gene annotation as the main data type we were going to put out, and then build up to more complex analyses like the comparative data. And for anyone who uses Ensemble, all of the views are kind of quite similar, so you'll feel at home in terms of what you can see there. We've done some minor quality of life updates, including things like in the species list, for example, making links available directly to things like the annotation files and the protein sequences and whatnot for people who want to do local analyses. But all in all, it's quite a similar experience. And just to give some more sort of information on rapid release, we're currently on 7 or 8 releases, depending on who you ask. We're targeting a sort of a 2-week release cycle, and we're more or less hitting that. We're going to work towards being more stable in terms of it being exactly 2 weeks, but we're more or less hitting 2 weeks at the moment. And we have already 139 annotations since launch. I mean, that's actually quite a lot of annotations. It took on the main Ensemble site many years to reach that count, and so it's quite, it's showing that we're able to get things out much quicker. What we provide now is the gene annotation, also repeat features, protein domains, an FTP site with all the different file dumps, and also a BLAST service. But what we're working on at the moment is also incorporating homology data into it, gene name assignment, and REST access. So the final thing I want to talk about is improving annotation. So the main thing is annotation isn't a solved problem. You only need to look at human and mouse, for example, where we have teams of expert manual annotators, both in Ensemble and RefSeq, working for years, and they're still improving the annotation in human and mouse, and they have an absolute wealth of data, and they're still finding all sorts of cool new things. You go outside of human and mouse, and there's a handful of model organisms that have really high quality annotation in the eukaryotes like Drosophila and C. elegans, but again, not very many. And outside of those, most species have very little data in comparison, and automated annotation is really the only annotation we have. And the quality of automated annotation is all kind of based around alignment quality and, the strengths and weaknesses of alignment in general. And so it's very heavily dependent on transcriptomic data, because the better the transcriptomic data, the more straightforward it is to do the alignments, and sort of the more representative it will be of the genes that are in the genome. But it's rare that we get very high coverage transcriptomic data, because it's expressed in different tissues at different points in time. So we still need to do a lot of work through homology data, and that kind of breaks down the further you move from a well-annotated reference that you're using for the homology data, the worse the alignments start to get. But the good news is things like isoseq and nanopore data, can give us much more confident full-length transcript structures. Short-read transcript reconstruction methods like scallop and string tie2 are continuing to improve short-read reconstruction. Family-level reference species are planned for a lot of these big projects where there'll be a lot more short-read and long-read data available, so the automated annotations will be higher quality. And then that, coupled with just higher levels of sampling per clade, means that comparative annotation is really going to come to the forefront over the next few years. But there's also huge potential to leverage like extensive sort of data sets in Ensemble and other resources to apply machine learning techniques to annotation. And just a couple of examples of this. Gene simple assignment, for example, we're working on, can we take HGNC, VGNC, MGI, ZFIN assignments, for existing species, combine that with, feature extraction from BLAST and structural properties of the underlying genes, and feed this into a neural network and use it to effectively come up with a gene name assignment for a new gene that we annotate. A nice other example is Camargo et al. Just recently released this paper and a tool called RNA Samba to help with assessing protein coding potential. And assessing protein coding potential is a difficult task in species that are distant from sort of a reference proteome, and particularly if the proteins are fast evolving, alignment-based methods are generally not sensitive enough in all cases to validate that something's actually protein coding or not. And this RNA Samba approach is a machine learning based method, and it looks like it's really very accurate, and it uses more kind of the intrinsic properties of the sequences themselves. And that's just a couple of examples. There are loads of other potential ways we can improve annotation through machine learning. So to summarize, there are lots of genomes to annotate. There will continue to be lots and lots of genomes to annotate. The annotation pipeline in Ensemble is continuing to scale well, and we're focusing on how can we scale deployment, to match that scaling in annotation. There's a lot of promising opportunities, particularly by machine learning, to both improve the quality of annotation and also make it more scalable. So with that, I'd just like to thank the entire Ensemble team. And yeah, if you have questions, please ask.
All right, thank you for this nice talk, Fergal. And I see we have a couple of questions coming in. I'll start with this one from Rob Waterhouse. This rapid release annotation is great. How does the pipeline incorporate legacy data, e.g. manual curation performed by community researchers?
So to be honest, it at the moment doesn't really incorporate community annotation unless that has been sort of deposited in some way as sequences into the archives, like historical cDNA style sequences. So we don't really have a direct method of easily incorporating community annotation, but it is something that we're actively looking at, particularly in the sense of, looking at making it easier for communities to create things like GFF files of their annotation, to sort of maybe submit to a validator for potential sort of future incorporation in our gene sets or as alternate tracks effectively in our sets. We also support, in general, we support track hubs. So we potentially have a route into the browser via track hubs. So track hubs are relatively easy to set up. You can register them in the track hub registry. And basically that's another route into the browser. But we are looking at kind of how can we improve working with communities to get sort of good manual annotations in. And actually, one very recent example was the update to Tasmanian devil, working on sort of MHC genes, incorporating annotation from the community for the MHC, where the automated annotation didn't do a fantastic job. We've kind of imported the old annotation for that and improved the annotation in that region.
Well, thank you, Fergal. I think we need to move on to the next talk. It's going to be given by Terence Murphy, who is the head of RefSeq at NCBI. He will be talking to us about annotation in the time of third generation sequencing. Thank you, Terence.
Hi, everyone. I'm Terence Murphy. I'm one of the team leads for the NCBI RefSeq project. Today, I'm going to give you a whirlwind tour of RefSeq and some of our resources. RefSeq's seen tremendous growth over the last 20 years. Now includes over 200,000 genome assemblies from prokaryotes, viruses, and eukaryotes. That includes over 17,000 named species and over 1,100 eukaryotes. Of these, nearly all of the genomes from multicellular eukaryotes are annotated with our in-house pipeline. Over half of those are new annotations or assemblies in the last 3 years across a wide range of species. We can't annotate everything at this point, so we prioritize mammals and species of biological, evolutionary, or economic importance. We prioritize better assemblies and those with community interest. So contact us if you want to get your genome bumped up in our queue. We annotate genomes to improve the utility of the public archive data, aiming to improve high-quality, consistent reference data sets to support research. Our pipeline's highly automated, almost entirely evidence-based. It integrates alignments of transcripts, proteins, and RNA-seq data to produce models that are optimized for full-length proteins and consistent with the available transcript evidence. We can use long-read transcript data from IsoSeq or Oxford Nanopore, but to date, we haven't found this to be enough to substitute for the read depth possible with Illumina. So for most species, we require short-read RNA-seq. All of the models are then categorized, named, and run through an orthology process that computes one-to-one orthologs versus a reference genome. The data is then released, automatically flows into all of our resources here at NCBI, allowing us to process most genomes in under a week with only a couple of hours of labor. We want to produce high-quality annotations, so we're always looking for ways to improve and optimize our pipeline. So recently, we decided to run BOOSCO across all of our annotations. In a nutshell, BOOSCO assesses a gene set versus a set of HMMs that are expected to be single copy for that species. It classifies the HMMs as being found as either complete, fragmented, or missing, and for the complete models, says whether they're single copy or duplicated. It's most commonly used on unannotated genomes in combination with Augustus to generate a preliminary annotation, but it can be run on other datasets, including finished gene sets. Here's an example of what the data looks like across all of the bird genomes annotated in RefSeq. You can see many score exceptionally well, the median score of 98, the best at 99.8% complete. The assemblies with small contigs and scaffolds score poorly, and most assemblies from long-read data are scoring extremely well. Across all of our data, we currently have a median complete score of 98.2%, with the best annotation scoring comparable to manually annotated reference sets from human and others. Only 19% of annotations score below 95% complete, largely reflecting lower assembly quality. We can also do comparisons to annotations for the same assemblies available in other resources. We find that RefSeq is consistently outscoring the other datasets we've assessed so far, often with 5 to 10% better complete scores, and only 3 genomes in GenBank with annotations from JGI that are currently outscoring RefSeq by just a hair. Most of the differences in these are from missing genes. There's a lot we can learn from these comparisons. We'll be digging into the individual gene results to see where we can make future improvements for RefSeq. BOOSCO does have its limits. The HMM sets might be biased towards genes that are easier to assemble or annotate. In particular, it's not very sensitive at detecting fragmented genes. Comparing to an assay we have looking at alignments to reference proteins, for this set of carnivore genomes, we see that the best have 89 to 91% of coding genes with very high coverage alignments to SwissProt, and some genomes are substantially lower. This correlates very well with BOOSCO, but the scales are quite different. Consider this pair for an older dog assembly and dingo. BOOSCO suggests there are only 0.7% of fragmented genes, about 150 total, but the alignments say it's really 5 to 7% worse, or 1,100 to 1,400 genes are fragmented. This is where long-read assemblies are really helping us give consistent results. The notable weak spot that's remaining is for what we call low-quality annotations. These are models predicted with frame shifts or internal stop codons, but we think they're likely to be coding genes based on orthology and other heuristics. We annotate them as coding with indels to compensate, 1 to 2 base pair introns in the GFF, and this label low quality on the protein to alert you that there's an issue. They're frequently caused by indel errors in the assembly, as shown by these alignments, which is a known problem with PacBio. The best genomes have low rates, below 3% for mammals, below 0.5% for the insects, but we see many genomes with higher rates, including PacBio and ONT-based assemblies in red. And we're currently rejecting about 10% of PacBio assemblies because they have high rates and need more polishing. This problem is very hard to assay, but aim for a sequence quality score above Q40. So as I said, all of the data in RefSeq is highly integrated through NCBI tools. I'll give you a quick whirlwind tour to get started. On the web, NCBI Gene is your portal into gene-related content, including all RefSeq eukaryote genomes. It supports search by nomenclature and other identifiers, provides transcript and protein sequences, a link to orthology information into our genome browser, some publications with a link to add your own through a mechanism called GeneRef, and a link for you to provide feedback to help us make improvements on specific genes. Our ortholog pages are accessed from Gene, currently support vertebrate ortholog information, and allow you to explore the sets, generate alignments and download sequences, and explore the literature. Our genome browser currently supports over 900 eukaryote RefSeq assemblies. You can configure the view to show additional tracks, You can view annotation from GenBank or Ensemble in addition to the RefSeq annotation. You can add RNA-seq expression tracks. And if there are multiple assemblies, you can view alignments to those assemblies and project a view of annotation, if that's available, into this view. Finally, datasets is a new way to access sets of data, currently supporting genome and gene data. It has web, command line, and API tools to make it much easier to download genome data for anything from single assemblies to hundreds of genomes for both RefSeq and GenBank. It can also produce tables of gene information in a customizable format. This project is rapidly evolving, so give it a try. Let us know what needs improvement, what other features you'd like to see. That's all I have time for. I've covered a lot of ground today, supported by a lot of really talented developers and curators at NCBI. Particularly want to thank Vamsi Kadali for the Busco work, Francois Thibault-Nissen, who heads the Automated Annotation Pipeline Group. And I want to thank all of the submitters to INSDC and SRA for the tremendous flow of new genomes and RNA-seq data into the public archives, which is what really makes this project possible. And with that, I'll take any questions.
All right, thank you, Terence, for this great talk. We have many questions that have come in, and it's a little bit unclear whether they're addressed to Fergal or for Terence, but I'll start with this one explicitly for Terence. How are you tackling annotating long non-coding RNAs, as these are poorly conserved across different species and not highly expressed?
Yeah, link RNAs definitely are a great challenge. Our pipeline does currently make an effort to stitch together link RNAs from short reads. It will also take advantage of the long reads when they're there. Our current approach aims a bit on the conservative side for representing these. So there's definitely more work to be done there. I think one of the biggest challenges is just knowing what is truth, and figuring out how to computationally get there.
Right. Another question from Anne McCartney that was asked earlier, actually, in response to Fergal's talk. It's really highlighting a need for the funding of, oops, that just skipped out. The talk is really highlighting a need for transcriptomic data as part of reference genomic efforts. Is this being considered by consortia?
I think my understanding, most of the consortia, at least the projects that are happening so far, both from RefSeq and I know from Ensemble, we've both been pushing for groups to be doing transcriptomic in addition to genomes, because it's really vital. I didn't have it on my slides, but we've also looked at Busco scores versus older RefSeq annotations, and haven't fully explored it, but it really, it shows what you expect to be the obvious, that adding transcriptomic data had really massive effects on improving annotation quality. So, and in space like especially like mammals, we can kind of do okay. That's like what Fergal was saying, where we have good understandings from homology. But when we get off into the great unknown, especially in the invertebrate space, we're really dependent on transcriptome to be finding genes.
Thank you, Terence. Unfortunately, there are many more questions, but we have to move on. So the next speaker today is Bastian Fromm. He's coming from the Department of Molecular Biosciences at Stockholm University and the Werner Gren Institute SciLife Lab at Stockholm as well. And he will be talking to us about microRNA prediction and MER machine. Thank you.
Hello, everybody. This is Bastian Fromm from the Friedlander Lab in Stockholm. I'm going to talk to you about our new pipeline MER machine that uses covariance models based on manual curated databases to annotate microRNAs. So microRNAs were discovered in 1993 in worms, and these are short hairpin-derived important gene regulators. And for some time, like 7 years actually, it was Sleeping Beauty, as shown here, and then discovered by more or less 4 labs independently that these microRNAs are not only found in worms, but actually are conserved in all animals. So when we look at them here, you can see the structure of 3 microRNAs in worm, Drosophila, and human, and also an alignment of a microRNA. You can see that literally across these clades, like 600 million years of independent evolution, sequential changes are very few. And indeed, microRNAs are very well studied and actually understood. You can see on the left a graph of how many annual publications deal with microRNAs. That is 12,000 last year. And on the right, you can see that the structure and the naming and how the processing actually works is also well understood. Despite this, the available microRNA complements that are found in Mirbase and the public repositories are very, very heterogeneous, and it leaves question that this actually is true or if this is actually a technical bias here. Now, I'd say there was another sleeping phase for the microRNAs until some of us, including me, actually tackled the problem. And you can see here in the bottom the number of microRNAs for these organisms after we re-annotated them. And you can see that very closely related organisms, pretty hard to read, I guess, here's human and macaque, they're very similar. And now we have actually a base to use microRNA complements for predictions. And we are also part of RNA Central now, which is great for us. Now, first thing we did is asking, how do microRNA annotations change between genome assemblies? And you can see here that microRNAs are actually lacking in some of these early drafts, meaning that microRNAs can be used to measure genome assembly quality. Also, and this has been extensively shown by other groups and us, microRNAs are excellent phylogenetic markers. And when you can see, you can reconstruct tree of life with it, but these purple dots here, you can see these are the question marks in the phylogenetic tree. And actually, microRNAs could be used to tackle them. And there are so many genomes out there, and of course, many more coming, and very few of them have been annotated for microRNAs. So that led us to the idea of using what has been implemented in RFAM, the RNA family annotation webpage, where you use covariance models that take into account the sequence conservation and also the structural conservation as depicted here. The lighter it is, the more structured. Now, what you do is you build models for each family in MirGeneDB that is more than 1,000, and we implemented this in our pipeline Mir machine here. I'm not going to go into the details. Essentially, we built models for every single family, and then looked at how accurate are these models predicting microRNAs. And there are a few outliers. I'm not going into the details here. Now we address them by looking at them more closely. However, when we look here, the number of copies as a heat map function of particular microRNA families across the tree of life, then you can see there are some that have a lot of copies, others that don't have so many. When we look at the predictions from Mir machine, this is without a priori knowledge, they are literally indistinguishable. Now we thought, let's use our model and try out the mammals that are found in Ensemble and annotate those genomes. And we did this right here. We annotated more than 30,000 microRNAs in one afternoon, and you can see they're very consistent across the tree, and we can even predict here in gray false negatives or possible microRNA pseudogenes that can be checked later. And also we see some genomes that probably are incomplete, would be interesting for Ensemble, I guess. And finally, when we included the MirGeneDB species here, you can see they're literally indistinguishable. As summary and conclusion, we have here a nice pipeline that is Mir machine that uses bona fide microRNA annotation for MirGeneDB, and we can accurately predict microRNA complements. And these will be then implemented in our next release for MirGenDB 3, and we are planning to release Mir Machine as pip and conda package by the end of 2020. Thanks for your attention.
Thank you, Bastian, for this interesting talk. While we wait for more questions to come in, is Mir Machine available as a component that can be plugged into another pipeline?
Yes, thanks. Great question. We're actually currently working on this. We're planning to have it finished by the end of 2020. The idea is to provide it as a tool that can be very easily implemented in existing pipelines and also as a standalone tool.
Great, that will be very useful. Question for you from Anonymous. What kind of data assemblies can you annotate microRNAs from? Great talk.
Thanks. So essentially, as I showed for the genome quality, it's very important that you have a high quality assembly in terms of completeness. But based on the complete genome, you need an assembly. So any FASTA, 2-bit, or whatever format you prefer works.
So I'll follow up quickly on this. Are there any particular parts of the taxonomy that you've had more difficulties with than others?
Yes, nematodes, because nematodes are really different when it comes to their microRNA distribution, really. There's a lot of losses, there's a lot of heterogeneity in the sequence and the structure. So they are a little bit challenging.
Mm-hmm. All right, thank you. Unfortunately, we have to move on again, but keep posting the question in the chat and the speakers can respond, I believe, a little bit later or even during the talk. So our next speaker is Yuantong Ding. She's joining us from the China National Gene Bank and will be talking to us about this resource. Thank you, Yuantong.
Hello, everyone. I'm Yuantong Ding. I'm a research scientist from China National Gene Bank. It's a great pleasure to be here. And today I'm going to introduce some of our featured databases, and especially one of the databases we developed for the COVID-19 virus. In that database, we incorporated with some new technologies like blockchain and secure multi-party computation to help promote the safe sharing and utilization of data. So first, let me say a few words about China National Gene Bank. CNGB came into service in 2016. It's a nonprofit institute and funded by the Chinese government. It's also the first national integrated gene bank in China. So the Chinese National Gene Bank is composed of 2 platforms and 3 banks. The 2 platforms are digitalization platform and the synthesis and the editing platform. Those 2 platforms are used for read and write the biological information, especially the DNA sequences. And the 3 banks are for the storage of biological samples, living organisms, and the bioinformatics data. And the CNGB database serves as the external portal for all the resources that we have at China National Gene Bank. So the CNGB database is built for biological data sharing and the application service. Currently, we have accumulated over 2,700 terabytes archived data. About 1/4 of them are public data. Some of them are controlled access data. And for the biological samples, we have collected about 500,000 samples, which covers over 2,000 species. We also built 20 specialized databases. Here is an overview of all the scientific databases we have here. It covers plants, animal, microbiomes, virus, and also there's a disease and health related database. Some featured ones are, for example, the 1,000th plant genome database and the 10,000 genome database for birds. And for the microbiome database, it contains the most complete data set of human gut microbiomes. And because of the limited time, I'm going to focus on the virus database here. So the virus database is we built this year for the COVID-19 pandemic. We are an official partner with GSAT initiative. As you may know, GSAT has been promoting the data sharing of virus data for over 10 years now. This database, we provide access to the most complete collection of COVID-19 genome sequences. Currently, we have about 0.1 million sequence data. And besides the virus genome data itself, we also provide analysis tools in the database. So if you click on the tools part here, it will take you to a computation platform. So here you can perform the fine-level genetic analysis with the virus genomes. For the analysis part, we provide 3 different modes. So you can do the computation by yourself. You can invite other users to perform the joint computation. And we also provide a third mode. It's called secure multi-party computation. So what's special about this multi-party computation is, so the computation actually happens at 2 separate computation nodes, and there are no plain text data exchange between those 2 nodes. So the users, they can store their data separately on 2 different nodes, and they don't need to send their data to the other part. So in this mode, it helps the user to have more control over their own data. And also we use the blockchain to keep the data records and the computation records. Blockchain, it's a distributed and tamper-proof system. So all the records on chain, they're tamper-proof. And we track all the data flow on the blockchain. But this is still an ongoing process. And if you're interested in blockchain or other cryptographic techniques, you could contact us for further discussion or collaboration. Thanks. Now I can take questions.
All right, thank you very much, Yuantang. That was a very interesting talk and lots of resources there. So one of the questions that's come in, it says, wasn't aware of the China National Gene Bank. Are you coordinating and sharing data with the other global gene banks? Amazing to have one more available.
Yeah, we do plan to, but China National Gene Bank, the database is built in 2018, so we are really quite new. So we are trying to build more connection with other databases, and as I mentioned, the GSAT is one of our steps. So we will build more cooperation with other databases, but we are mostly focused on our national resources here currently.
Okay. So another question for you, for me, how fast is your virus database growing? You said there was something like 125,000 sequences in there towards the end of September. How quickly is that growing?
Like today, I think it reaches, there'll be like 20,000 more in 10 days.
Okay. Okay. Yep. One of the questions actually mentioned the number in GSAD now, so I think you've already answered that question. Okay, great. Thank you again for the talk. I think we are ready to move on to our next speaker, who is Monica Polchow. from the National Agricultural Library at the Agricultural Research Service at the U.S. Department of Agriculture. And she's going to be talking about facilitating data reuse for genome-enabled communities. Please take it away, Monica.
Hi, I'm Monica Polshaw. I work for the USDA ARS National Agricultural Library, and today I'll be talking about facilitating data reuse for genome-enabled communities. The recommendations that I'll present here are based on our experience directing an insect genomics database, but it's also based on groupthink from many different colleagues and collaborators who I'd like to acknowledge before I start. And they're listed here. You'll likely see variations on these recommendations elsewhere. These are that I'm presenting today are tailored for the biodiversity genomics community. The Earth Biogenome Project will generate massive amounts of reference quality data sets with the goal that these data sets be reused across the scientific community and substantially drive scientific discovery. In order to be reused, the data need to be available in public databases. Now, there are many different kinds of databases which specialize on different data types and different community needs. These databases need to share data amongst each other, and scientists, or the data consumers shown right here, need to be able to access these data from the different databases. Finally, with large amounts of data, the sharing needs to be efficient. In the next slides, I'll introduce 3 sets of recommendations that both data producers and database providers should consider to enable this data reuse. The first recommendation is to follow community standards or create them as a community if they don't exist yet. So what do I mean by standards or best practices? First off, there are file formats, for example, the GFF3 format for genome annotations. There are metadata standards, for example, MIGS 5.0 for genome metadata. And there's another example is nomenclature standards. For example, if in your taxonomic community, there may be specific ways of representing gene or protein names. Now, these sound like very straightforward things to adopt. In our experience, it's actually challenging to adopt all of them and do that well. So our basic recommendation for databases on this front is to join or follow a group that leads standards development or implementation. One example here is AgBioData. I list a number here. At the end of this talk, I'll have a slide that will show links and resources to all of the recommendations that I post here for your reference, and you should be able to access these later. For data producers, we recommend that you generate genome project data with submission to the appropriate database in mind and find out early what the submission standards are. Second of all, rich machine-readable metadata is critical for data reuse. So metadata is information about data. For example, if you generate a genome assembly, the FASTA files would be your data, but the assembly programs and other information about that process would be part of the metadata. Metadata is crucial for scientists other than the data generator to understand and reuse the data set. And description of the data set in a publication is not sufficient. Often it's behind a paywall, so not everyone can read or access that. And it's not machine readable. So if a scientist wanted to access all these data sets at scale, that would not be possible in terms of the metadata. So what do we recommend? For databases, it can be challenging to retrieve the metadata from data submitters because it's often an afterthought. So it can help to provide incentives to get metadata. For example, if you get an excellent metadata set from a user, provide more services for them. Second of all, we recommend to use ontologies or controlled vocabularies to the extent possible. EBI's ontology lookup service is especially helpful. For data generators, we recommend that you embrace your metadata. So especially for these reference data sets for the Earth Biogenome Project that are tailored towards reuse, think of metadata as an essential product that you're also generating to enable reuse. And record your metadata as early as possible and provide as much as you can. Here are some useful resources for this that I provide links to as well. And finally, plan and budget for software development, operations, and maintenance. Sharing data requires software that stores and serves it, and biologists, I'm guilty of this myself, often underestimate the cost that it takes to develop, but also especially operate and maintain software over longer periods of time. So for databases, we recommend that you use and contribute to existing open source software if possible, for example, Gmod. Follow best software development practices. Budget, good developers are expensive and can be challenging to recruit and retain. Build for web services and dial in on user needs to reduce software bloat and costs. And finally, for data generators, we recommend to keep this in mind when you're reviewing grant proposals for databases. Thank you.
Okay, thank you very much, Monica. That was a very good talk. As we wait to see if some questions come in, I have one to get started on. So these lessons that we've learned so far, what do you think are the most important things going forward that basically the lessons we haven't quite learned yet? Where do we need to be focusing on next?
Well, in my talk, I address what I think are the top ones. I think that for this audience of the biodiversity genomics community, it's a diverse set of people. We've heard talks today from the larger databases where obviously some of these issues aren't that big of a problem. My talk was addressed to both data generators and also large databases, but smaller ones and ones that might be generated as we obtain more genomic resources and other genome-enabled communities might produce more smaller databases. I would guess the metadata would be the big hitter for all of these to really think at data generation and when you're generating your database itself as well, to really focus on getting rich machine-readable metadata. We've all heard about the FAIR data principles, I presume, to make data findable, accessible, interoperable, and reusable, and especially with these large reference data sets that are supposed to be a strong resource to enable research across the community. It's imperative that we have enough data to describe them and that it's machine readable. And so thinking across the community about metadata, I think is the biggest priority.
And do you see a role of kind of a community, basically a community workshops to drive those metadata?
Yes.
And how do we bring in everybody that we need into those meetings?
Yes. So I guess I can throw a shout out. I said this during the talk as well, or in one of my slides, that the Earth Biogenome Project has an IT and informatics subcommittee that we will be releasing our recommendations for the project soon. And look out for that. And we'd appreciate feedback on that as well, as we have various iterations of those standards. But there are other existing communities that are already working with existing genomes. I referenced the Ag Biodata Consortium, where we're heavily working on facilitating metadata access and reuse for our scientific community.
Okay, great. Thanks very much. Okay, our last speaker today is Scott Edmonds from GigaScience Press, and he's going to talk about a new publishing workflow for rapid dissemination of genomes. Take it away, Scott.
Hello, greetings from GigaScience in Hong Kong, where I am going to talk about the very end of the whole biodiversity genome kind of creation process. Obviously, this meeting has been covering, how genome projects have kind of gone to a planetary scale. In the last decade, from Genome 10K, there's been a huge growth of these consortiums tackling the various different corners of the tree of life from a kind of taxonomic and geographic perspective. This has really been driven by technology, by scaling up of kind of genomics pipelines, starting from the technology, sequencing chemistry, automation, then to the data handling and kind of infrastructure side of that, which is about kind of maintained pace. The least attention has been to the very end of this workflow, the kind of dissemination process, which essentially hasn't changed that much in about 350 years, right? Credit is through publication, and it's not really been fit for purpose of these data sets. Since 2012, GigaScience has tried to address that specific problem on the cultural side through giving people credit through data papers, right? So these data note articles that describe a genome. And the one thing that we've done different is we also have our own open source data repository, GigaDB. You can download it and run it yourself. And this comes with in-house curators who will organize, structure, and make sure that nothing falls through the gaps. All of the key genomics data, if there isn't a home for it, there are no excuses. We've done pretty well, even if we say so ourselves, over the last 8 years. In our birthday in July, we have seen that we've published about 765 papers. About 230 of these are the specific data description papers from many institutions and from some of these consortia projects. The thing I'm most proud about is all of the data that we managed to liberate, about 46 terabytes to date, about 2,000 data sets, about 1,200 of these specifically genomic data. So in this kind of 8-year period, we've learned a lot. It's worked to an extent, we've had hundreds of these papers, but as things scale to thousands, tens of thousands, millions, there's some sort of lessons learned here that things that have kind of held us back and made things problematic have been the technology. The kind of publishing infrastructure is pretty terrible. It's slow, it's extremely expensive, the end products are static, and still everything's narrative focused. So we feel that the way to get around this is to follow the software paradigm: code, release, fork, update, and repeat. And decided to basically start working on a new publishing project, working with River Valley Technologies, who've built this amazing XML end-to-end pipeline. Having everything in one pipeline, it makes integrations a lot simpler. You don't get this kind of API spaghetti. And the advantages of this, doing everything in XML, means that doing everything in one platform means there's no import-export of files, faster, more accurate from that perspective. Cutting out the production will save a huge time and cost savings. It means that versions can be published near instantaneously. You can update, you can fork, you can push a button and spit out a typographic quality PDF, change the views, change the language, change the fonts. And through this technology, we've then decided to launch this new journal, Gigabyte, focused on forkable products to start with, your data, your software. We want to leverage embeddable dynamic content and make peer review easier with a kind of questionnaire. The first example that we've published has been this, the Banjo Frog from that was one of the first 101 G10K projects that's basically been orphaned for a decade. You can see that it's got protocols embedded in and features like this. So all in all, yes, thanks to all of these people and River Valley Technologies, submissions are currently free for the next 6 months.
Thanks, Scott. That was really cool. What do you think is the kind of unit that works best here? 1 genome, 10 genomes, 100 genomes, 1,000 genomes?
So it's how people want to use them. People have their own, they're interested in little corners of the taxonomic tree. They're working on specific conservation projects. I think genomes themselves are useful. The amount of narrative that you will get out of one genome is sort of decreasing. A lot of the interesting stories are already out there, and the bigger, more glamorous journals are focusing on these more multi-genome taxonomic. The reason people are doing these big studies are to answer these big phylogenetic questions. And so, there's scope for big narrative overview studies from that, but people still need the original genomes. You haven't done everything yourself. It will still help somebody, and you can't wait 5 years for a consortia project to finish to before all of this stuff gets released. So make incentives, make it easier, just reduce the kind of barriers to just getting that stuff out.
Here's one of the questions that's come in. Does GigaDB forward relevant data sets to international repositories, or do the authors still have to do that themselves?
So we won't send it out unless all of the data is available. That we look through for genomics is well looked after with INSTC databases. We'll check that at the beginning, make sure the authors have done this, and we can help them. We've got curators on hand who can kind of hold their hands and help them all of that, and we won't let it go out until all of that is available. But GigaDB is there to basically fill the gaps with all of the other stuff. We'll take the BoostGo files, we'll take results and things like that that are useful for a reproducibility perspective, non-standard intermediate data that may be useful. But yeah, we can help people get stuff into the INSTC, but we're not officially INSTC ourselves. So yeah.
Gotcha. Thank you, Scott. So this wraps up our session today. Thank you to everyone who joined us. Thank you to all the speakers who were part of the annotation and databases session, and to everyone who listened in to the stream. This is an incredibly exciting time. It's great to see all of these developments and things happening all over the place. We hope that you enjoy the last two conference plenaries, and look forward to seeing all of you next year at the next meeting. So again, thank you, and have a good day.